miscarriage and premature labor for women. And it wasn't realized until some years later that this actually has a teratogenic effect because it's mimicking some of the hormones in the developing fetus. And so when those babies were born, they were initially fine, but when they grew up to be adults, it was realized that their own reproductive tracts had some malformations due to the exposure to this as embryos. So, we don't use this anymore, obviously. Fetal alcohol syndrome, I'm sure you guys have heard of this. It's more common than you would think, even within the US, about 1 out of 650 children. And here are just a couple of brains. This is a newborn wild-type brain here, whereas A is someone that has been exposed to large amounts of alcohol in the fetus. And then these colored tracks are myelinated neurons. So it's showing you a measure or a rubric of neuron growth within the brain. And in C, there's far fewer of these, and they're a little bit less elaborate in terms of where they go to as well. So, sometimes a woman might be pregnant and might be having a glass of wine here and there without even realizing it. And the sort of conventional wisdom is that probably is not a problem. Generally with fetal alcohol syndrome, the mothers are either alcoholics or really drinking a lot of alcohol. But on the other hand, who wants to risk that? So, a lot of women would choose not to drink at all. Okay, here is just some more examples of embryos that have been exposed to mothers with high concentrations of alcohol. And in particular, some of the defects that you can see have to do with the craniofacial development. So within this developing mouse here, you're looking straight on at its nose and snout and mouth. And notice the size and structure and shape of these things. Then over here in B, the whole upper lip and palate area is much reduced in size. And they're also having, it looks like, some trouble with the neural tube closure. So remember, the neural tube has to, once it invaginates, has to eventually pinch off and seal into a tube that goes the whole length of the embryo. And then C, we've had appropriate neural tube closure, but the size of the head's a little smaller, and again, the size and structures of these craniofacial regions are not quite the same as in wild type. And then here, these two pink organs are the olfactory glands, and these are reduced or absent in the mouth with high alcohol exposure. So, this may not be lethal, but it still could be pretty severe. So what would be the mechanism of this? We're always trying to think about, not only can we just make this correlation of we observe something, but we would actually like to try to explain it in cellular terms. And one probable factor that's playing into this is the inducement of these superoxide radicals by the oxygen. So these are known as a group as the reactive oxygen species, or ROS. And here's just a couple of examples of things that would fall into that category. And they all have an oxygen atom in there. Oxygen atoms are critical for life, and they have these certain chemical properties, one of which is they're highly electronegative, and they really like to attract electrons. And, as long as they're in their traditional type of bonds, they're not going to be a super reactive type of atom. But if they get in different kinds of configurations, just the chemistry of it is such that it's really looking for another electron. And so it might steal one off of any kind of protein or DNA or any kind of macromolecule in the cell that it might come into contact with, and this can lead to downstream consequences. So in this scenario here, we have a normal embryo and an alcohol-treated embryo, and they're stained with a mechanism to look at cell death. And within the alcohol-treated, there's extensive death of just individual cells throughout the developing brain. So what you're looking at here is the head of an embryo before neural tube closure. So down in the center, here's the neural groove, and then the neural ridges to either side. And look at how many black cells are cells undergoing cell death here. And then to really make sure that we're interpreting this correctly, you can add into the experiment now an inhibitor of the reactive oxygen species. So we let these be present, but we block them, and then that's sufficient to ameliorate the cell death, so that goes away, and you get more of a normal development. So this kind of experiment where you make an observation and then you specifically block one step, it helps us to be more confident that what we think is happening is actually happening. So it's an experimental support for the idea that ROS molecules are actually causing cell death and that's damaging the embryo. Yeah?
Where do these reactive oxygen species come from, like specifically from the alcohol?
So the alcohol, it's a good question, and I don't know a lot of detail about the answer, but the alcohol itself is a fairly small molecule and has an OH group on it, which can be pretty reactive. So that may be just the source right there, is that it's not going to be just these individual molecules, but derivatives of the alcohol itself that are interacting biochemically in all these processes. Okay, so a little bit, one more slide on this is that in embryos that have this fetal alcohol syndrome, another thing that we can see is that the cells within the embryo are not as adhesive to each other, so they're not sticking together as well as they really should be. And so we have in D there 3 representations of cells. These are probably cultured cells taken from embryos. And in D, you can see they really want to adhere well and clump together. With a smaller level of alcohol, there's somewhat more dispersion, and with higher levels of alcohol, even more dispersion. And then on this graph here, they're looking at the ethanol concentration from 0 to 50 millimolar, and then how well those are adhering. And it's less adhesion as the concentration of alcohol exposure goes up. And then L1 is a particular receptor that normally should be mediating this, so if the cells are not expressing the receptor, then they don't show the same reaction. Okay, so then we can also have exposure of the mother to different types of radiation or viruses, and this can cause teratogenic effects. But one of the things is rubella syndrome, which is a virus, and it's German measles is what it is. And so if the mother has not had this and she contracts it while she's pregnant, then that can have teratogenic effects on the embryo. And the kinds of things you see are somewhat smaller head, deafness, heart deformity, or eye deformity. And again, think back to that chart of what are the organs that are developing first in the embryo at the earliest time. It's all these things: the central nervous system, the heart, then the eyes, and brain shape. Okay. So oftentimes, if there is examples of pollution or other factors, it can lead to some effects on organisms developing in the wild, such as tadpoles. And here's an interesting example where you've actually gotten the body duplication phenotype. So this may trigger some ideas in you about what might have gone wrong in that embryo. Maybe the development of the nucleus center was affected, maybe the Spain organizer is affected, something during gastrulation started out okay and didn't go quite right. And so you can find certain pollutants that will do this, but also just heavy metals such as zinc, mercury, or lead. They themselves, if present in the proper concentration, can lead to some defects that are affecting gastrulation and body patterning. So those are all external types of things that may happen to you. Exposure to the wrong compounds, exposure to heavy metals, different things out in the environment. But then the body itself has signals that it's sending around as part of this process of embryo development. And one of these is retinoic acid. The body makes this partly using vitamin A as a source. And normally it's distributed in the embryo in a gradient. And so you actually see higher concentrations of retinoic acid toward the head, and this tapers off as time goes by. One thing that retinoic acid can do is to turn on and off the Hox genes. So that's going to be a pretty powerful tool to try to manipulate body patterning, because if you change the expression of Hox genes, you're going to get a lot of impact on the developing anterior-posterior axis. So here's a couple of experiments looking at this, and they've taken embryos at early stages and just soaked them in excess retinoic acid. And the somewhat milder version of what can happen are the craniofacial defects. So in this embryo here we have this dark staining element here, which is part of the jaw. It's called Beckel's cartilage. And this is much more rudimentary in the panel D, where the embryo is treated with excess retinoic acid. And then E has been a super treatment, a really large excess of retinoic acid, and it's even gotten to the point where it now is affecting anterior-posterior patterning, where the head structures have become predominant within the embryo, but then the trunk and the tail structures just never developed. So I would expect that the Hox genes that should have been turned on in that area maybe were never turned on due to this manipulation by retinoic acid. Okay, any questions? All right, let's go ahead and take a break. It's 8:45, let's go to 9:45.